What's up, guys? It's Gary with Fresh from the Farm Fungi. I'm here in Denver, Colorado in my mushroom cultivation lab, and today I wanted to do a simplified video on how to breed mushrooms, specifically mushrooms for production. So I guess there's 4 basic principles when you're breeding mushrooms, and if you haven't seen our videos that go really into depth on all these principles, check out our playlist, breeding mushrooms from spores. It goes into really detail about this, but someone requested me to do a brief overview of the breeding process, so I'm going to do my best to kind of simplify all of that into one short video, and then you can go through all the rest of the videos and look through, I guess, more examples and the exact sterile procedures that are required in order to breed mushrooms successfully. So the first principle in breeding mushrooms is to isolate the spores. So in nature, mushrooms will drop its spores after it has achieved its peak growth state, and before it gets consumed by insects or animals. It wants to reproduce just like any other organism, so it does this by releasing spores. So you can capture mushroom spores on a surface like a piece of paper, tin foil. I prefer to use sterile petri dishes, so I have a bunch of these lying around. Usually when they get like cracked a little bit, or if I use a culture completely, I'll take the agar out, clean it, and then I'll have a great place to make a spore print. So essentially you just take the mushroom and then you'd cover it with something to keep the humidity high enough where it can release its spores. It does that through hydrophobic pressure, and it will release its spores onto the surface. So you can think of the spores as almost like a seed for a plant, but that is the most important thing when you're breeding mushrooms. You have to have a spore, or you have to have multiple spores, because typically Basidiomyces will fuse together from separate spores to form mycelium. So mycelium can be thought of as the roots of the mushroom. It's actually just the complete mushroom, but that's the easiest way to think of it. So principle 1 is obtain and isolate spores. So you can isolate spores using a few different techniques. I have an example of a serial dilution I've been working on for some piapino, but essentially you want to be able to take this massive quantity of spores, which it could appear as a tiny little speck on a piece of paper, but microscopically you have to separate those spores. So thinking of it as, you know, if you're planting seeds in your garden, you don't want to take a whole bag of seeds and stick it in one hole. You want to take one seed and plant them out evenly. So the whole purpose of that is to be able to isolate one specific trait from that spore. So you can do that by streaking. There's a method called a streak plate, where you would take a sterile loop, streak it out on some agar, and basically you're spreading those spores using physical means. The other method, which is the serial dilution, you can take a diluted sample of spores and continuously increase the volume per spore until in one little vessel you have maybe 1 to 10 spores. Then you take a drop of that solution, put it on a petri dish, and then you have a single spore isolate. There are other methods. I guess you can isolate them physically with some kind of a cell counter, or another method that has been used traditionally for cordyceps mushrooms is to take a fruiting body, attach it to the lid, and collect the spores in real time as they're being dispersed from the fruiting body. So that is the first principle in breeding mushrooms, is you have to isolate a single spore or a few spores that are isolated from each other So then I guess the next principle is to cross those spores. So the reason you isolate them is so that you can combine them together. So I guess this is probably the most confusing part in breeding mushrooms, is how the spores cross with each other. So if you're doing a streak plate and you have a high condensation of spores, there's a chance that as soon as they germinate and produce a hyphae, that they'll instantly fuse together with another spore. So it's very important to isolate them so that as they grow out a haploid hyphae or mycelium, which is half of the genetics, so it's a gamete cell. It doesn't have the full genetics. I mean, there are some exceptions, but essentially you want to take half the genetics, let that grow out, take the other half of the genetics, let that grow out, and combine them together. People get confused when they streak out spores, or if you take a really dense volume of spores from a spore print and then inoculate agar with them, because there are some colonies that will form that have mated microscopically. So this can be confusing because you can take those colonies and they will not fuse with another haploid. They're already mated. So there's a process called sectoring, which I can get into in a different video, but essentially that is going to let all these spores kind of fuse together randomly on a single petri dish, and then you're observing them and searching for the strongest growth, or I guess it's just mostly by chance or choice what sector you prefer to isolate. So the second principle of breeding mushrooms is going to be crossing those spores. So you can do it really systematically, like in the videos that I have provided on YouTube, or you can do it kind of at random by doing a streak plate or taking a spore solution and putting it onto a petri dish. Or PF Tech is a really powerful tool where you can take a spore syringe, inject it into a 3D matrix, and let the spores compete against each other, and then I guess the strongest will fruit out, and then you can clone those. So principle number 2 is crossing the spores. So principle number 3 is going to be to grow out those crosses. So that is a whole nother process. I have tons of videos on how to grow mushrooms. You can follow from spore to fruit, and basically every different species of mushroom is going to have its preferred method to grow. So in the wild, it just selects a specific niche in the environment, whether it's going to be on decaying matter like the compost loving secondary decomposers, or if it's on hardwood like an oyster mushroom and it would land on a dying tree in the wild, germinate, seek out another spore that has landed, and then they'll fuse together and form mycelium. Then the mycelium will grow out in the tree and form a fruiting body, and that right there is breeding in the wild. So it's a miracle that this happens every day in nature, and it's really hard to replicate this in a sterile environment in the lab. So I guess once you have your genes crossed, there's not really a way to verify that without using genetic testing. So a single haploid mycelium will look very similar to a mated pair. So the only way to know for sure is to grow out and select the strongest genes. So that's going to be the 4th principle, is growing them out and selecting for the best genes. So this is an opportunity where you can select mushrooms for your own growing environment. So here in Denver, Colorado, we're at a higher elevation. So it makes more sense for me to breed my own strains because the levels of oxygen are going to be less just being at a higher altitude. So I found out that some of the strains that I've been trying to grow out from my mentor in New York, they just didn't do as well because of the elevation difference, which kind of forced me into breeding my own strains to cultivate. So oxygen consumption, CO2 limitations, these are all traits that you can breed for. Another example would be different matrices or different substrates that you can breed for. So say you're in a remote area and you don't have access to hardwood fuel pellets, you can breed mushrooms that will favor like different agricultural waste or straw. So these are different factors you can select for, which would be the fourth principle of breeding mushrooms. So I'll go through that. Isolate spores, cross the spores, grow out the crosses, and then isolate the strong genetics and clone those. So cloning, I guess, is another process in breeding, but it's kind of separate on its own. I have a few videos on how to clone mushrooms if you want to check those out. Some of the key factors or key features of cloning are going to be sterility, genetic diversity, and daily observation. So these are, I guess, the hardest things to account for during the whole process of breeding. So sterility, if you don't have either the species that you're attempting to clone, or if you get different molds or bacteria in your sample, it's going to ruin the whole process. So it has to be sterile, which is why I prefer to do the serial dilution technique, because by the principle of using serial dilutions, even if you have contamination, it's going to be mitigated as you dilute your sample. There are other ways to achieve sterility. You can grow your mushroom in vitro, which means it's inside of a vessel, and then that way it's not exposed to the outside environment. Streaking out the mushrooms on a petri dish will also somewhat achieve sterility. You can observe if there's contamination, but the problem with that is that you can get other types of mycelium that looks similar to what you're trying to accomplish. So I've done that in the past where I tried to breed lion's mane, and then just by chance there was an oyster spore in that plate, and I ended up growing out oysters, which, I mean, that's not the worst thing in the world, but when you're trying to breed a very specific mushroom, it's important to keep that in mind. So then the next topic of discussion, or I guess one of the questions I get asked, is about the genetic diversity. Can you use like a monoculture, take those spores and breed them? And the answer is yes, but at a certain point it is going to produce worse and worse quality fruits over time. So it's important to build a diversity in your breeding. So take mushrooms from different areas of the environment, or different cultivators throughout the country have different genetics, and it's better to use a wide assortment of genetics so it keeps that genetic pool big. Otherwise, you're going to be selecting for very specific genotypes over time, and eventually it's going to create problems. But the quick answer is you can breed from the same monoculture, but the long answer is it will have consequences over time. So then the last part is probably the most important part. Maybe sterility is more important, but daily observations. So if you're not observing your petri dish every single day, then you're going to miss contamination. If you have two mushrooms in grain spawn and one of them does really good in 3 days, but then kind of stalls, and then the other one grows faster, then you're going to miss selecting for speed. There are a lot of observations you can make as a cultivator that are going to determine the success of the breeding process. So every single day you have to observe, write down your observations, and when you're selecting for what mushroom you're going to clone, it's going to come in handy rather than going through everything haphazardly and just picking a random mushroom. So I hope you guys enjoyed that video. I tried my best to cram it all in one quick video. Give us a thumbs up if you enjoyed this video. Subscribe if you're looking forward to more mycology videos like these. I've got some new shirts on our Etsy, so check out our Etsy Fresh Fungi. We also have our live cultures available, and we started shipping internationally too, so check out and see if we can ship to your country. Etsy has been, tremendous help for our company to help us distribute all of our different genetics. So I really appreciate you guys, and I hope you enjoyed this video. Give us a thumbs up, and until next time, much love.